Hello biologists, in today's daily biology revision task you are asked to produce a flashcard on the effects of inhibitors on the rate of enzyme controlled reactions. Right, as you can see from the graph on the right hand side, without inhibitors the rate of reaction is considerably higher than with. You need to remember there are competitive inhibitors as well as non-competitive inhibitors. Reversible inhibitors can bind to enzymes through weak non-covalent interactions such as ionic bonds, hydrophobic interactions, and hydrogen bonds. Because reversible inhibitors do not form any chemical bonds or reactions with the enzyme, they form rapidly and can be easily removed. Thus, the enzyme and the inhibitor complex can be rapidly dissociated in contrast to an irreversible inhibitor. Irreversible inhibitors covalently bind to an enzyme which causes the chemical changes to the active sites of the enzymes and cannot be reversed. The main role of irreversible inhibitors include modifying key amino acid residues needed for enzymatic activity. Reversible inhibitors bind non-covalently to enzymes, and many different types of inhibitor or inhibition can occur depending on what the inhibitors bind to. The non-covalent interactions between the inhibitors and the enzymes include hydrogen bonds, hydrophobic interaction, and ionic bonds. Many of these weak bonds combine to produce strong specific binding. Competitive inhibitors, as the name suggests, compete with the substrates to bind to the enzyme at the same time. The inhibitor has the affinity for the active site of the enzyme where the substrate also binds to. This type of inhibition or inhibitor can be overcome by an increase in the concentrations of the substrate which outcompete the inhibitor. Uncooperative or uncompetitive inhibitors bind to the enzyme at the same site as the enzyme's substrate. However, the binding of the inhibitor affects the binding of the substrate and vice versa. This type of inhibition cannot be overcome but can be reduced by increasing concentrations of the substrate. The inhibitor usually follows an allosteric effect where it binds to different sites on the enzyme than that of the substrate. The binding to an allosteric site changes the conformational shape of the enzyme, so therefore it will no longer bind to the substrate. Non-competitive inhibitors bind to the allosteric site and reduces the activities but does not affect the binding of the substrate. Therefore, the extent of the inhibition depends on the concentration of the substrate. Hopefully, this video should have helped you complete your daily revision task. Please subscribe so you can keep up to date with other videos to help with your daily revision tasks. You can follow me on Facebook or follow me on Twitter to help you keep up to date.